Computational biology is the science of studying biological mechanisms using computational approaches. I'm leading the group of Computational Epigenomics and Systems Cardiology, where we on the one hand use machine learning on large data sets to extract information that we can use to understand the large system that there is in human cells which are connected to diseases. How can we use large data sets that are created to understand which genes cause a disease and which mutations are relevant? Modern technologies can measure so many aspects of our body down to the molecular level, and piecing these things together is one of our missions. Our focus is to study cardiovascular diseases, and many of our projects revolve around the center of ourselves, the human heart.
The Uniklinikum project was funded by the BMBF to develop an energy-optimized programmable chip that is able to detect heart arrhythmia. Imagine a really small chip that can be placed on your smartwatch or even in your body, and it's able to detect these heart abnormalities that is commonly known as cardiac arrhythmia. In the same way that a doctor detects arrhythmia by ECG measurements, we have developed a deep learning model that can classify them automatically. This is a joint work with a group of Professor Hedrich. We have developed energy-optimized components, automatic chip design, and a segmented deep learning model in order to minimize the chip area. Diagnosing arrhythmia is really important, but to understand the mechanisms behind it, we have to look at the cellular level.
So when people think of the heart, they usually think of a huge muscle. But actually, the heart consists of different cell types. Besides cardiac muscle cells, we also have cells of the vasculature, immune cells, parasites, mesenchymal cells, and many, many more actually. By using microscopy or histology, we are able to specifically label these cells with antibodies and visualize them. So indeed, by using histology, we can validate and confirm the findings that colleagues of us have made bioinformatically. So in the lab, you can manipulate cardiac cells by using an ex vivo system, the so-called heart slices system. So these are slices from real human hearts that were explanted. In the lab, you can use then different stress factors. So you can add different beating frequencies, you can use different currents to set the whole system under stress. We currently also use the SARS-CoV-2 virus and added this virus to these human heart slices. And indeed, we found that the virus was replicated, the heart muscle was infected, cells even died, and basically got some disease-like structures. We're also dealing with different mutations and virus strains and variants, like the virus from the UK, from Brazil, South Africa. These mutations are not restricted only to the viral genome. Also in humans, we have DNA mutations. These mutations can then cause different kinds of diseases, such as cardiovascular diseases.
A small change in the DNA might sound unimportant, but can still have a huge impact. Most of the observed DNA mutations are not located in a gene, meaning they do not change the gene sequence itself, but how a gene is regulated. This is because a large part of the DNA functions as regulatory regions for genes, and these regulatory regions can increase or decrease the activity of a gene. And we applied machine learning on a huge collection of datasets to construct a database containing 2.5 million regulatory regions. And this database, called Regio, is publicly available, and anyone can look for the genes of interest and the interacting regulatory regions.
By using our database Regio, we try to find genes which are affected by the mutations. Our method helps to prioritize DNA mutations in order to identify the impact on human diseases. Doing so, we identified candidate disease genes in arrhythmic patients that likely explain the difference in the ECGs in comparison to healthy people.
What we have shown here was a glimpse on the complexity that we and others are facing to solve these current problems. Computational biology is a fast evolving field. New technologies are being developed, and large international consortia produce gigantic datasets that every day create new paradigms for the analysis that we need to solve. While we're constantly getting better at zooming in into the very molecular detail, so much remains to be discovered for us. If you want to work at the intersection of biology, medicine, and computer science, come and join us.